Hello everyone, my name is Kunal Kundu and I am a PhD candidate at the University of Maryland, College Park in the US. In this tutorial, we will learn to look up variants in VCF files and further annotate the variants with genomic region, allele frequency in population, mutation type, predicted impact, and association with genetic diseases. This is one of the primary steps towards variant interpretation. For this tutorial, we will analyze a dummy VCF file generated from ion torrent sequencing data. Let's open the VCF file and analyze the format. Here, I have opened it using Excel as the VCF file is small. However, in real life, one operates on these files programmatically because of large size. The VCF file has a header section where the lines starts with a hash and a variant section containing information about genetic variants. From the header section, we learn about human genome version used for variant calling. In this case, it's AG19. Variant caller software used. In this case, it's Torrent Variant Caller. Definitions about various quality control parameters such as DP, total read depth at the variant position, and genotype information. The first 9 columns in the variant section, as highlighted in red, indicates chromosome at which the variant appears, coordinate of the variant, ID indicates an identifier of the variant, such as identifier from dbSNP database. Ref indicates the reference allele at the position. Alt indicates the mutant allele at the position. Qual indicates the variant quality score. Filter indicates if the variant has passed all the quality filters. If yes, gets a pass tag, and if not, the failed quality filter name appears here. Info indicates different types of variant quality annotations defined in the header. Format indicates the genotype annotation sequence. The next column has the sample ID as the column header, under which the genotype information of the sample for each variant is recorded. Now that we briefly understand the structure of a VCF file, let's try to answer some questions about a variant on chromosome 1, position 155408635. As a result, I will demonstrate the usage of the information included in the VCF file to answer these variant-specific questions. Question 1. Is this variant present in the given VCF file? The answer is yes, as highlighted. Question 2. What is the reference allele at this position? The answer is T. Question 3. What is the mutant allele at this position? The answer is C. Question 4. What type of variant is it? That is, single nucleotide variant, insertion, or deletion? The answer is single nucleotide variant, as there is a single base substitution here from T to C. Question 5. Is the quality of the variant good? The answer is yes, because it has a pass annotation under filter column. Question 6. Is the variant homozygous or heterozygous? The answer is homozygous, because in the genotype info column it is 1/1. The number on either side of the slash indicates the presence of the type of allele on 2 copies of the chromosome. 1 here refers to the mutant allele and 0 would refer to reference allele. 1/1 indicates that the mutant allele is present on both the copies of the chromosome and therefore homozygous. If it was 0/1, we would call it a heterozygous variant. Question 7. Is the genotype quality good? The answer is yes, it is good, because the GQ, that is 105 value, is greater than the threshold value of 30. This threshold value varies from different variant calling software, and one can figure out the value by going over the software's documentation. Now, let's move to the other aspect of the tutorial, that is variant annotation. There are many tools available for variant annotation such as ANOVER, SNPF, VEP or Variant. In this tutorial, we will use web server version of ANOVER called WANOVER. It is one of the popular tools for annotation purpose. Here is the WANOVER website that provides a form to submit the job for annotation. We will start with filling an email ID to which the results will be sent. Next, enter the sample identifier in the VCF file and then upload the VCF file. An important step here is to appropriately fill in the annotation parameters. As we noted earlier in our dummy VCF file, that it is generated using Human Genome version HE19, we would select the same here. The input format is VCF. The gene definition file can be set to refseq gene. However, you may use other gene definitions for a more comprehensive annotation. All other parameters will be set to default, and finally click Submit button. Upon successful submission, there will be a link to view the results. The results page provides options of downloading the annotations in a comma-separated file as well as TXT file, and you may also view the results in the browser. The result page also distributes a separate file for variants in the exonic region of the genome. Let's view the genome summary results in the browser. This page displays a large table that contains variant annotations. Let's parse the information in the table by asking clinically relevant questions about a variant. Let's consider a variant on chromosome 7, position 40498,796 for this exercise. Question 1. Where is this variant located with reference to coding region of the genome? As noted in the func column, this is an exonic variant. That means it's part of genic region of the genome. Question 2. Which gene hosts this variant? In the gene column, it displays the gene name as SUGCT. Question 3. What kind of mutation does this variant cause in the protein? As noted in the exonic func column, it causes a non-synonymous mutation, also known as missense mutation. You may look at the amino acid change column to know the mutation position and amino acid change in the protein. Question 4. Is the variant novel, rarely or commonly observed in human population? To answer this question, we would refer to databases such as 1000 Genomes and EXAC that contains information about the allele frequencies in human population. According to the column 1000 GAll, this variant has very low minor allele frequency of 0.0006, and based on column EXAC-Freq, The minor allele frequency, that is 0.0056, is low as well. This indicates that this is a rare variant. Question 5. What is the predicted impact of the variant? There are many computational methods available for variant impact prediction, but in this tutorial, let's focus on two popular ones, SIFT and polyfin. According to the columns, polyfin 2 HDivPred and polyfin HVarPred, the predicted impact is damaging, indicated by letter D. For this tool, benign is indicated by letter B, and possibly damaging is indicated by letter P. According to the column SIFTpred, the predicted impact is damaging, indicated by letter D. Benign is indicated by letter T in SIFT prediction. Question 6. What is the dbSNP identifier of this variant? dbSNP is a database of single nucleotide polymorphism maintained by NCBI. It collects variant information such as minor allele frequency from many sources such as 1000 Genomes and Exact. Here is the database website. Back to the annotation table. Based on dbSNP column, this variant has ID RS137852860. Now with this RS number, we can query dbSNP website to collect information about this variant. Question 7. Is this variant previously known to be of clinical significance? To assess such information, let's go over to the ClinVar database, a freely accessible public archive of reports of the relationships between human genetic variants and phenotypes with supporting evidence. We can query ClinVar with the variant's RS number. The interpretation section of ClinVar annotates this variant as pathogenic and is associated with glutaryl-CoA oxidase deficiency. The evidence of this assignment is provided in the assertion and evidence details section. The evidence section indicates that this variant has been reported pathogenic by 3 submitters. This brings us to the end of this tutorial, where we have learned about how to parse variant information in a VCF file, how to annotate genetic variants, and further interpret variants by putting together the annotations. Thank you for watching the tutorial.